Everyone likes to be fashionably late at webinars. All right, so maybe we're going to make a start. Thank you very much again to everyone for attending that webinar organized by Streck and Everest Biolabs. So my name is Dimitri Aubert, and I'm the Vice President for Sales and Marketing at Everest Biolabs. And today I'm going to co-present, but the talks are going to be presented by Yunjun Song, who's Senior Scientist, Research and Development at Streck, and Tal Gilboa, who's co-founder and Head of Research at Everest Biolabs. And the presentation will be about reproducible EV workflows from blood. So I'll leave the floor to Tal first. Here you go.
Do you hear me?
Yes.
Thank you, Dimitri, and thank you everyone for joining us. So I'll just start with a quick intro to extracellular vesicles, which I'll refer to as EVs in this talk. Wait, are the slides moving?
No, there's still the presentation.
Okay, cool.
Yeah.
You see it? Okay, so EVs are nanoparticles which are released from all the cells in the body. And as they are released to the blood, on their surface, they contain markers from the cells of origin. And as they are leaving the cell, they're capturing whatever is inside the cell. And so they have cargo of molecules like RNA and proteins from the cell. And so they carry this very big set of biomarkers and have potential for diagnostics of multiple diseases. However, using EVs for clinical studies is challenging because of the different steps which are needed to conduct EV work. So first, there is the plasma or blood collection. So for blood collection, you will hear a lot in Unju's talk after me, where the main challenge is how do we really maintain the EV distribution when we collect the blood and when we store it, and keep the EV stabilized. Then after we collected the blood, we need to isolate the EVs. And there we have challenges of how to do it with high yield, high purity. We have a background of lots of other molecules, which can be very high concentrations like protein, lipoprotein. And after we did do that, how to do it in a reproducible, high throughput, automated manner. And then after isolation, we need to use these EVs as biomarkers. So we have the analytic steps. And also there we want to have assays which are sensitive, specific, can detect low and high levels of EVs, and have high throughput and simple. And so I will focus on these two parts of the EV work. And for isolation, the reference method for isolating EVs from blood is SEC, which is separating the EVs by size. And the way it works is we have these columns, and we fill the columns with resin, and the resin has small pores. And then we add the plasma on top of the columns. And so the small particles, like the proteins, will interact with the resin with the small pores inside, while the large particles, like the EVs, will just go through the column and elute first. So if we start collecting fractions, we will have the EVs, which are here in green, eluting in the early fractions, and then the small proteins will start coming in the later fractions. And this method is really great for EV isolation because we can fine-tune the yield and the purity of the EVs by changing the resin in the column. It's a very gentle method. We don't apply high forces on the EVs, and so we keep the EV integrity, they're functional, and have the same characteristics as they started with, and it's amendable to automation and throughput. And so at Everest, we developed a few SEC columns, which are specifically useful for plasma and serum research. We have them in different formats. So you can start with low input volume, of up to 300 microliter. You can go with a typical plasma volume of 0.5 to 1 ml. And there is also the high throughput version, which is in a plate 24 well format, which acts just like an SEC column. And the thing we really care about is the reproducibility, because if the columns are packed differently, you will have the EVs eluting earlier or later in different factions. And so we have very strict QC criteria to have the EV elution be the same across our columns. And then we have 3 different flavors of columns, which are different by the yield and purity of the EVs you will get. So for the 6B apex column, it has the smallest pore diameter, and so it has the highest yield. You get the majority of the EVs after the isolation. But for plasma, you will have coelution of lipoprotein and some leftover free proteins. Then we have the apex 4B. It has a larger pore diameter, so it cleans the EVs better. You have very high depletion of albumin and small proteins, and also lipoprotein, specifically high-density lipoprotein, which is smaller. But you have slightly lower yield than the 6B. And then for those who are working on untargeted methods like proteomics, who want really clean EVs and making sure the particles after isolation are mostly EVs and not lipoproteins, we have the apex MM columns. And the way they look, they have 2 different layers. So on the top layer, it's basically an SEC column. It separates the EVs from the small proteins by size. Then the EVs reach the bottom layer when there is another cleaning step. So it further depletes lipoproteins from the EVs, and you end up with very clean EVs, where the majority of the particles detected, and here you can see some NTA data, are actually EVs. So that was the isolation. And now after you have your EVs with different purity and yield according to your application, typically you need to analyze them. So there are 3 main methods currently used to quantify EVs after isolation. People are using particle counting, which is a very strong method. It can count the EVs to get a quantitative concentration relatively fast. You can get single EV resolution using nanoflow. However, if you don't do a labeled particle counting, which is more complicated and needs lots of optimization, you are basically measuring particles. So you can't differentiate an EV from a lipoprotein. And in blood, lipoproteins are much higher abundant than EVs. And so you do want to add some specificity to this measurement. So Western blot can do that, where you can really target a specific protein of interest in your EVs. You can have second validation by the size, so you know you're detecting the correct protein with Western. However, you cannot distinguish if this protein was on an EV or it was a free protein. You have limited sensitivity, and it's very hard to run Western on plasma because you have the smear of all the other molecules. And so the other method which can complement is ELISA. It has another layer of specificity because you have now a sandwich assay with 2 antibodies. There are very sensitive ELISAs with light dynamic range, so you can detect low and high levels of your protein, and it works in complex biofluids like plasma. However, again, if you're just looking at the protein with ELISA, you don't know if it was on EVs, and some workflows are laborious and involve coating of the plates, multiple washes, which is time-consuming and laborious. And so we developed a very fast single-step EV ELISA. And the way it works is basically you add your sample, and this ELISA works with either regular biofluids, so plasma, serum, CSF, urine, or with EVs after the isolation. So you add your sample to a strip. You can run 1 strip at a time, up to 12, so up to 96 samples. Then you add our reagents, and our reagents are antibodies against the tetraspanins, CD9, 63, and 81. You have 1 incubation step, We do not burst the EVs during that step. And then you have one wash step. And then you add the substrate, stop it, and detect. So it's a very fast assay with just one wash step, and it works with biofluids or purified EVs. And we have two versions. In green here, you can see the absorbance assay, which is a very fast and simple assay, but has limited sensitivity and dynamic range. And so to extend that, we also have the Lumi, which is chemiluminescence based, which is what Unju will present in her talk. And this assay can detect around 10 times lower EV concentration, and it has a 4-log dynamic range. So you can measure in the same time samples with high amount of EVs and low amount of EVs with a single dilution factor. And you can also conjugate your own capture and basically colocalize a specific EV transmembrane protein with tetrasphenins. So just example for that, we used CD41 to capture EVs. We took plasma samples and fractionated it before and after platelet activation. And then we measured the CD41 assay as CD41 as capture and tetrasphenins as detector. And you can see how after the activation, we have higher amount of CD41 EVs, but we can detect both the high levels and the low levels in the same run because of this 4-log dynamic range of the assay. And so I showed you our isolation, columns, analytics. We also have instrumentation to automate the isolation, which I did not present today. But again, our goal is to just advance diagnostics and therapeutics development and support clinical trials using EV biomarkers by addressing all the different isolation and analytics parts of the EV work. And I will pass it to Unju, who will focus on the pre-analytical sample collection step.
Thank you, Tara.
Thank you.
All yours, and you.
Yeah. Let me see. Can you see my slides?
Yes.
We can.
Okay, yeah. Thank you. Thank you for inviting me to this conversation. I'm Unju Song, a senior scientist in R&D at Streck. I've spent the past few years working on pre-analytical variables in blood-based EV research. We were one of the early and enthusiastic adopters of Atlas Lumi human EV ELISA assay. For some time, we had been looking for a sensitive and robust quantification tool to measure blood EVs reliably in blood samples, while distinguishing true biological signal from pre-analytical artifacts introduced by sample collection and handling. Lumi EV ELISA delivered exactly that. Building on what Tal has just explained, I want to frame this EV story from the angle that drives our interest at Streck. EVs are membrane-enclosed particles released by virtually every cell type. And what makes them powerful as biomarkers is that they carry molecular cargoes from their cellular origin, messenger RNAs, microRNAs, lipids, and protein. These molecular signatures reflect physiological states in real time, including health and disease states and treatment responses. Because EVs circulate in blood, they offer a window to distant organs, making conditions like cancer and neurodegenerative diseases accessible through a simple blood draw. Companies like Mercy Bioanalytics and Mersla Bio have already introduced EV-based blood tests for early cancer detection. Mercy's Mercy Hallow for ovarian cancer, Mersla's Evo Livo for liver cancer. So the translational momentum is real. But to get there, the field has to solve a technical problem that sits upstream of any assay or biomarker, which is pre-analytical variability. Before we begin, let's review what blood is. Blood is a complex matrix composed of red blood cells, leukocytes, platelets, and plasma with proteins, lipoproteins, and EVs. To analyze EVs, blood cells must be removed first. And there are two fundamentally different approaches. Serum is generated by triggering decoagulation pathway, but the clotting itself activates platelets, releasing non-physiological EVs. And clotting time varies among samples, which is another potential confounder. Plasma avoids clotting altogether by using an anticoagulant and provides a more controlled starting point. But the anticoagulant choice matters. EDTA and citrate-based tubes activate platelets to different degrees. And centrifugation speed, rotor type, and break settings all influence EV composition in your final plasma fraction. This means EV populations are sensitive to the plasma isolation step itself, and making standardization more challenging and more critical than for other analytes like cell-free DNA. Why is this happening? Imagine blood in the body, in dynamic circulation, at steady temperature, with tightly regulated gas composition. But the moment blood is collected into a tube, all of that changes. In response to that abrupt ex vivo transition, a cascade of events is triggered. Platelet activation, red blood cell chlorination and homolysis, and leukocyte degranulation. Each of those events releases blood cell-derived EVs into the plasma. The important thing to understand is that this process is progressive. It starts immediately after the draw and continues over time. If you process your sample right away, within an hour or so, you can capture something very close to the in vivo state. But if processing is delayed, those artifactual EVs accumulate, and the signal you measure increasingly reflects the collection and handling artifacts than the in vivo biology you are trying to capture. Standard collection tubes like EDTA or ACDA were not designed to stop these events. They manage coagulation, but they do not inhibit cellular activation, blood cell breakdown, or EV release. This brings us to one of the most important but underappreciated decisions in EV study design. Who isolates the plasma and where? In the liquid biopsy field for cancer, there is an established standard. Whole blood is collected into stabilization tube at the clinic and shipped to a central laboratory for plasma isolation. One lab, one SOP, one set of trained operators. That's the model major liquid biopsy companies use today. But in the EV field, there is no consensus. Many groups isolate plasma at the blood collection sites and ship the plasma to a central lab for analysis. This decentralized plasma isolation model can introduce operational complexity. Each site needs its own validated protocol, its own trained operators, and its own audit trail. Assume you have 10 collection sites. Your regulatory burden has multiplied by 10. Centralized plasma isolation eliminates that. But it only works if you have a tube that keeps whole blood stable during ambient shipping. The EV field has been exactly asking that question. In 2023, Ann Hendricks' group at Ghent University published the first blood collection tube benchmarking study in collaboration with EV researchers across Europe. They systematically evaluated blood collection tubes, including ACDA, EDTA, at a few processing time points, measuring the impact on various EV quality metrics, including residual platelets, homolysis, red blood cell-derived EVs, and platelet activation. Their conclusion was direct and clear. Both tube type and processing interval significantly impacted EV performance metrics across most metrics. The authors specifically called for further research to develop a blood collection tube that preserves draw-time EV concentrations and molecular signatures for longer periods to enable clinical implementation. That study gave us a clear picture of the scope of the pre-analytical problem and a concrete set of benchmarking metrics. So what does a blood collection tube actually need to do for a translational EV research? The requirements are clearly laid out. It needs to preserve draw-time EV concentrations during delayed processing. To achieve this, it needs to control platelet activation and limit residual platelet contamination. It needs to minimize hemolysis and the red blood cell-derived EV artifacts. Importantly, it needs to do all of that without interfering with downstream EV isolation and analysis platforms. We evaluated Nucleic Acid Plus BCT, the newest blood stabilization tool from Streck, against these requirements. Using Atlas Lumi human EV ELISA kit from Everest Biolabs, we examined the stability of total EV concentrations. Blood from healthy volunteers was collected into EDTA, ACDA, and Nucleic Acid Plus BCT. Whole blood assigned for delayed processing were stored at ambient temperature for up to 6 days. Right before plasma isolation, tubes were inverted 5 to 10 times to mimic agitation during blood stripping. Then plasma was isolated by a double spin protocol, 1800 G for 15 minutes, followed by 2800 G for 15 minutes with full break. EV concentrations were measured directly from plasma after 1 in 40 sample dilution using the Lumi EV ELISA kit. Thanks to its large dynamic range, this single dilution factor was sufficient across all plasma samples, which became really handy when dealing with hundreds of samples in large batches. So let's look at the data. In EDTA and ACDA, EV concentrations increased progressively with processing delay. By day 3, EDTA showed about 10-fold increase relative to draw time, and ACDA about 8-fold. By day 6, ACDA reached more than 40-fold. In contrast, Nucleic Acid Plus remained close to draw time at both time points, on average 1.7-fold at day 3, 2.4-fold at day 6. Among all these samples, I want to highlight one donor with unusually low baseline EV concentrations across all tube types. In Nucleic Acid Plus, that donor remained stably low throughout the entire 6-day window. In ACDA and EDTA and ACDA, the same donor sample rapidly increased and became indistinguishable from the rest of the cohort by day 3. That tells us the ex vivo EV increase is driven by the blood collection environment, and Nucleic Acid Plus better preserved the true biological range across donors. The biological range becomes confidently visible only when collection and detection are both optimized, which is exactly where our presentation titles come from. Collection meets detection. To understand the mechanistic basis of those bulk EV changes, we used single particle flow cytometry to measure blood cell-derived EV subpopulations specifically. This analysis was performed by EV count in Amsterdam using an apogee flow cytometer with lineage-specific markers for platelet-derived EVs using CD61, for erythrocyte-derived EVs, CD235A, for leukocyte-derived EVs, CD45. Note that this data set includes Streck RNA complete BCT, which is a predecessor to Nucleic Acid Plus as an additional comparator. What we found was consistent with the EV ELISA data. While ACDA and RNA complete showed substantial increases over time, Nucleic Acid Plus kept all 3 blood cell-derived EV subpopulations close to their baselines. We also measured residual platelets by flow cytometry. In ACDA, platelet concentration increased about 22-fold by day 3, reaching about 8,000 platelets per microliter, enough to meaningfully contaminate downstream EV analyses with platelet-derived artifacts. We also observed substantial donor-to-donor variability in residual platelets and in blood-derived EVs as well, suggesting not all donors respond to the same degree. This matters because depending on when plasma is isolated and from whom, the sample matrix shifts in ways that are difficult to anticipate or control, making population-level standardization challenging. By contrast, Nucleic Acid Plus maintained platelet concentrations at baseline through the delayed processing window. Next, we looked at EV subpopulations with more translational relevance using MaxPlex EV Immuno-Oncology Panel from Mateni Biotech. This kit simultaneously profiles 37 EV surface markers from a single plasma sample using antibody-coated beads and flow cytometry. Briefly, plasma samples were incubated with the beads overnight and washed, then stained with APC-conjugated detection antibodies and run on a flow cytometer. With this kit, we were able to monitor compositional shift in many cell type-specific EV subpopulations. Here is the MaxPlex data. These line graphs show fold changes relative to day 0 for 37 EV surface markers at day 3 and day 6 across EDTA and EDTA, ACDA, and Nucleic Acid Plus. For visualization, I grouped these markers into 8 cell type-based functional categories. Looking at EDTA and ACDA, platelet-derived EVs show the largest divergence from day 0, reaching up to 100-fold in platelet EV case. Although to a lesser degree, the divergence is systemic across all the categories. The direction and magnitude vary by markers and by 2, telling us the composition of EV population is shifting, not simply scaling up. In Nucleic Acid Plus, which is marked in gray, most markers remain close to their day 0 values throughout both time points, indicating the integrity of draw time EV composition is better maintained in Nucleic Acid Plus. The final analyte class I will cover is RNA. EVs carry messenger RNAs, microRNAs, and other non-coding RNAs. EV-associated RNAs have attracted substantial interest in liquid biopsy as complementary analytes to cell-free DNA. In this study, EVs were enriched from plasma immunomagnetically using stem cell technologies EasySep Pan-EV kit targeting CD9, CD63, and CD81. EV RNA was extracted with the Promegas Maxwell microRNA plasma kit. We quantified 3 microRNAs by real-time PCR, MIR165P, LES785P, MIR1038-3P, as well as ubiquitin B messenger RNA by droplet digital PCR. The data show clear tube type differences. In EDTA and ACTA, EV-associated mRNA UBB increased about 30 to 35-fold during delayed processing time, compared to 2 to 3-fold increase in nucleic acid plus. MicroRNA story was similar. Progressive increases in EDTA and ACTA, with nucleic acid plus maintaining more stable signals throughout for all three targets. Let me summarize what we have shown. We examined nucleic acid plus BCT across 5 EV quality metrics: bulk EV concentration, residual platelets, stability of EVs derived from blood cells, and EV subpopulation profiling, and EV-associated RNA. Nucleic acid plus was found to help limit hemolysis, platelet activation, and residual platelet contamination. It helps maintain EV population integrity, EV RNA integrity during delayed processing. We also confirmed compatibility with ELISA, single particle bead-based flow cytometry, and EV RNA analysis. Additionally, although not shown here, we confirmed compatibility with several EV isolation methods, including size exclusion columns from Everest Biolabs. So what does this mean for how we design our EV studies? Regardless of tube choice, plan your pre-analytics before you lock your assay protocols. Define the blood collection workflow upfront: tube type, storage and transport temperature, processing window, centrifugation settings. Validate tube compatibility with your specific EV isolation and detection method. Keep in mind the tube choice affects residual platelets, blood cell-derived EVs, EV-associated RNA, and EV surface marker profiles. Consider all analytes of interest before you commit to a blood collection and plasma isolation workflow. With that framework in mind, where does nucleic acid plus BCT fit? It adds value in specific contexts. Multi-analyte studies requiring EV, cell-free RNA, and cell-free DNA from a single blood draw. Decentralized or multi-site collections where delayed processing is part of the study design. Prospective translational research studies where intersite reproducibility is a primary requirement. So my final message for you: pre-analytical standardization should not be an afterthought. It is foundational to your assay, to your data, and your path to the clinic. Thank you for listening. I hope this has been helpful. We'd love to hear your feedback and support your journey of biomarker discovery and translational research workflow optimization. Please reach out to us through the Streck Early Innovators Program or our email, stabilization@streck.com. Now, I'm happy to take your questions.
Thank you very much, Yunju. Very nice presentation, and to you, Tal, as well. Yes, the floor is open for questions. If I'm trying to.
Hi,
yeah.
Nicole
Hello,
Harris in the Fluid Biomarkers group at Biogen. My question is around RNA isolation kits to isolate EV RNA. You mentioned the Promega Max RNA kit was used in looking at the effects of those pre-analytical variables on EV RNA, but did you compare?
You're cutting, Nicole.
Sorry?
No, we hear her.
You can hear me? Yeah.
Yes.
Okay, my question is if you compared other RNA kits to compare like the EV RNA yield and quality across different kits.
So we did, but not very extensively. So I remember the Promega's Maxwell EV the kit that we used, a Promega Maxwell microRNA plasma and serum kit. Their IFU says that it's compatible with RNA isolation directly from plasma as well as EV, but your EV should be diluted in or eluted in water, not PBS. So definitely something coming from PBS may interfere your EV kind of RNA binding or extraction. So kind of I think if you think about plasma versus EV, the kind of salt and all the compositions are very different. So extraction kits, how they deal with all those different matrices is another problem. So definitely, I think that's another step. That's another kind of thing that we need to or not optimize, but compare and then see how extraction kits affect EV RNA yields.
Thank you.
Oh, yeah, I see the question. So you're asking if the columns can be used on a cell culture media? So definitely yes. The columns are validated on cell media with and without concentration, CSF, plasma, basically almost all biofluids, milk, EVs. So yes. Okay, I can do.
Hello. Sorry, I had a problem with my microphone. I just sent you the question. Did you say that EVs should be diluted in water and not in PBS? And I know I've seen that PBS shows up like in NTA or SEM analysis, but water only? Doesn't that cause lysis of the EVs?
Oh, so yeah, so kind of to clarify, that's kind of when you want to extract RNA. So of course, yeah, you don't want to kind of elute EVs in water. If you want to have the intact EV kind of for your downstream application, but yeah, when you extract RNA, you want to kind of break the EVs, right? So in this case, if you my workflow was EV isolation was kind of immunomagnetic capture of EVs and then isolating RNA from those captured EV. So in that workflow, if you add water, it doesn't matter much because you are going to lyse anything. You are not going to lyse EVs anyway. So in that particular case, I mean, I need to actually double check it now. Kind of,
yes,
I
please.
think that it was water, not PBS. And then I have another kind of kit has a compatibility issue with because of this buffer with a Promega Maxwell microRNA kit.
Okay, thank you very much.
And then kind of the background there is that, RNA, kind of EV RNA are small. So those to capture those small RNA, those are very sensitive to the kind of binding matrix, kind of depending on the salt composition and everything, how those tiny RNA will be captured to whatever your resin is a very kind of delicate process. So kind of it definitely the matrix, the binding, the binding environment. What I mean is like RNA binding to resin, that environment is very kind of delicately, should be very delicately controlled to maximize RNA recovery.
Absolutely. Thank you very much.
I see Nicole is asking about CSF. So yeah, Nicole, I've done a lot of CSF work in my postdoc, which I'm happy to discuss offline. Typically, we use between 0.5 to 1 ml of CSF on the regular apex columns. With the 6B, you get the majority of the EVs, and you can also further concentrate them using ultrafiltration. So the yield with the 6B is quite high. Yeah, and I'm happy to share with you CSF data. We also have it published. Yeah.
And there's a question from Benedetta Bessolati about for you, June. Did you compare your Nucleic Acid Plus with the conventional Streck tubes in terms of nucleic acid and maxplex analysis?
So conventional means, I guess, I assume they are referring to our kind of cell-free DNA too. So I'm trying to kind of remember. Oh yeah, we have a comparison data. So if you want to see kind of course, kind of cell-free DNA is designed to stabilize white blood cells only. But yeah, this nucleic acid plus BCT, our design concept is like stabilize all the cell types. And when you think about EVs, red blood cells and platelets are the major sources of blood cell-derived EVs. So controlling those things were critical. So that's why kind of compared to Streck cell-free DNA BCT, nucleic acid plus it kind of shows much more improved performance regarding keeping EV concentrations stable, as well as cell-free RNA, because most cell-free RNA, especially messenger RNA, are packaged inside EVs.
Okay, and I see a great question from William regarding the how do we convert the data into a particle count? So that's a great question. What we do is we provide with the kit a standard, which we characterized. So we characterize it with nanoflow and other methods. And we assume a typical biofluid number of tetraspanin per EV. So we correct for that because the cell called the standard we provide has more tetraspanins than usual per EV. And so you have that as a reference. If you're using samples which are not plasma or biofluids like cell culture, you can also use your own standard for this conversion. But again, it's not like measuring a specific protein and using a calibration curve with that protein. So it's semi-quantitative in that sense. And we mostly use these ELISAs like Unju do to compare different conditions or look over time. And so for that, it's actually an excellent QC ELISA assay.
And comments are your question about receiving information about the tubes and the ELISA kit. We can send you an email after the event to give you that information from Streck and Everest.
And if you have other questions after the talk, you can feel free to email me, Unju, and Dimitri. Yeah.
No hands rising. Right, in that case, I think we're going to close the webinar. Thank you very much again for attending, and I hope you found it useful. Yes, as Tal was saying, don't hesitate to contact us afterwards if you have any questions. Thank you again. Bye-bye.
Thank you. Bye.
Bye. Thank you.